Hi, I'm a senior high school student in California, and over the summer, the last 10 weeks, let's say, I've been conducting a research project alongside my mentor, Dr. Agarwal, into the tumor suppressor gene TP53, apropos of a type of brain cancer called glioblastoma. Uh, as it has been for a lot of people, it's been pandemic season for the last year and a half. So, towards the end of the last spring, I had started to look for research opportunities that could be remote to enhance my learning, as the virus prevented us from doing in person or lab meetings, right? And I found just the right opportunity through SkoolMentor. Turns out that being remote also has the advantage of allowing learning between a student and mentor who are halfway across the world from each other, as was the case for me. So here's what I believe to be the most important takeaway I had from this research experience with Dr. Agarwal, and I'll take a moment to back up and provide some context for the itinerary of the project I went through. The stages we went through in my research project was first to refamiliarize myself with the biology 101 concepts, like how cells make proteins, what is the cell cycle, what are pathways, etc., and then to conduct a literature review of the papers existing on the topic that we were researching, to then utilize the database that we used to explore lacunae that we identified from the literature review, and finally, to wrap it all up in a presentation to a symposium. So let's talk about the second step there. From learning about glioblastoma in general to understanding specific pathways of chemical interactions in it, it was necessary to read through many papers on the topic. As you might imagine, scientific papers about super specific topics might not always appeal or are able to be easily read and understood by a layman, which I am. Therefore, it often required reading papers several times and using visual aids that often came from the papers and spending hours at a time on a particular series of 2 to 4 papers. And I would go through a stage of frustration and think that I didn't understand. But after a while, and usually around 2 hours, my understanding of what the paper was saying solidified greatly. A big part of it was looking up the terms from the papers and reading about them. But that sounds a lot more simple than you're reading through a paper, you don't understand a sentence, or the whole abstract, you've got to Google each part of the sentence and put it together in a way that makes sense, and then do it for the rest of the paper. Though, to be fair, some of the things would end up repeating themselves, and it wouldn't be necessary to re-Google everything that you looked at before. To some extent, when you look at a paper, and even sometimes reading its title, for example, characterizing macrophage diversity in metastasis-bearing lungs reveals a lipid-associated macrophage subset, you might think, what in tarnation does that mean? And my biggest takeaway from this program has been that it'll help overcome that obstacle of not understanding, because if you do it once, who's to say you can't do it again? Thanks again to SkoolMentor for providing me this opportunity to work with Dr. Agarwal.